Welcome to our lecture on Darwinian agriculture and plant breeding, an introduction. My name is Karl Schmid. I am a professor in the Institute of Plant Breeding, Seed Research and Population Genetics at the University of Hohenheim in Stuttgart, Germany. I am also one of the co-organizers of this summer school, and in this lecture I will give you a very high-level introduction into the topic. Hopefully, we meet in person at the course site in Montpellier. Let's begin with the question: why do we actually think about or discuss Darwinian agriculture? There are several reasons for this. Firstly, our croplands are the results of evolution. This means that all crops we cultivate today have descended from their wild relatives. These plants have been domesticated and further developed by humans over hundreds or even thousands of years, adapting to our current agricultural systems. In this context, we can view plant breeding as applied evolutionary biology. We are utilizing the principles of evolutionary biology to improve our plants, principles I will introduce shortly. The goal now is to apply concepts of Darwinian thinking in agriculture and plant breeding. You will see that these concepts differ slightly from other perspectives, such as a purely genetic view. However, it is important to note that genetics is of course an integral part of evolutionary thinking. By integrating these evolutionary concepts, we can enhance our understanding and practices in agriculture and plant breeding. My lecture has the following outline. First, I will introduce to you Darwinian and evolutionary thinking. Then I will show to you how this thinking is relevant for agriculture. We have two main learning goals for this lecture. First, you should understand the key concepts of Darwinian thinking and Darwinian agriculture. These concepts are usually not covered in the other courses or in any other courses, and by understanding them you should be able to formulate scientific questions in agriculture using the principles and concepts of evolutionary biology. To illustrate this, I will provide several examples showing how this type of thinking can be applied to agricultural settings and plant breeding. But first, let's begin with the term Darwinian. What is meant with this term Darwinian? Essentially, the concept of Darwinian thinking dates back to Charles Darwin's seminal book on the origin of species, which was published in 1859. In this book, Darwin presented the theory of evolutionary biology, describing it through a series of concepts. The first concept is that evolution is based on variation between individuals. When we observe individuals of a species, we see that they differ from one another in different characteristics or traits. Next, we have the inheritance of traits, meaning that the traits of an individual are passed down to the next generation. It is important to note that Darwin did not know how genetics worked. The mechanisms of genetic inheritance were discovered about 40 years later in 1900. Nevertheless, Darwin understood that traits could be inherited. Finally, Darwin identified that some traits are advantageous over others and leading to the selection by differential reproduction. Individuals with advantageous traits have higher reproductive rates and produce more offspring than those with disadvantageous traits. By combining these three concepts, variation, inheritance, and differential reproduction, we can formulate that any system meeting this criteria is an evolving system that changes over time. This framework has many implications and applications. For instance, we can develop computer programs that incorporate these principles, creating evolvable systems that exist only in the digital realm. For our purposes, it is crucial to understand that these principles apply both to natural and to artificial selection. Artificial selection, defined as selection by humans, is precisely what we do in plant breeding. By applying Darwinian principles, we can enhance our breeding strategies and improve agricultural outcomes. The importance of plant breeding in understanding basic evolutionary theory is highlighted here. This is the first page of Darwin's famous book on the origin of species. The front page introduces the concept of natural selection. In the first chapter, Darwin discusses variation under domestication. This discussion underscores that plant breeding, which includes domestication, a subset of plant breeding, as well as animal breeding, is crucial for comprehending the fundamental principles of evolution. By studying plant breeding and domestication, we gain valuable insights into how evolutionary processes operate and can apply this understanding to explain the mechanisms of evolution. By examining wild plants and their domesticated descendants, we can clearly demonstrate that domestication is an evolutionary process followed by subsequent plant breeding. This can be seen in the example shown here. Here we see the wild ancestor of eggplant, Solanum insanum. This plant looks quite different from modern-day eggplant, but you can already identify some key characteristics of the Solanaceae family, which are also known as nightshades. For example, you can see the typical flower structure of nightshades. Sorry, this doesn't work so well. The fruits bear a resemblance to those of the tomato, which is characteristics for the nightshades. Additionally, Solanum insanum exhibits several traits that are typical of wild plants. Notice the presence of thorns. Maybe it works with this. Yeah, the presence of thorns. And also that the fruits are highly toxic because they contain alkaloids that are produced by the plants. Following domestication, these plants have undergone significant changes, as you can see here. They now exhibit a variety of shapes, which are the results of selective breeding. Different genetic variants have been selected to produce desirable phenotypes, for example in the color or in the form of the fruit which we are eating. And importantly, these plants have become edible. We can eat them now because the alkaloids are not produced in the fruit anymore. So through the process of domestication, humans have selectively bred out the alkaloids in the fruit, making it safe for consumption. A very important concept in Darwinian agriculture and in evolutionary biology is selection, specifically natural selection. We can differentiate between two types of selection: individual selection and group selection. This distinction is crucial for understanding how differential rates of reproduction occur, as illustrated here. Keep in mind, differential rates of reproduction lead to a change in the phenotype of a population, as an advantageous phenotype will be increased in frequency in a population. In this picture, you see wild barley, the ancestor of our cultivated barley, growing at the Dead Sea, 400 meters below sea level in the Levant region. These plants thrive in highly saline soils with extremely high salt content directly at the edge of the Dead Sea. You can see here some individual plants in the picture. Here we can easily imagine in such an environment that selection occurs at the level of the individual. Each plant struggles for survival in this harsh environment, and those plants or genotypes that manage to endure the salty conditions produce more seeds than those that cannot. This scenario exemplifies individual selection, where the ability to survive and reproduce is determined by the specific traits of each plant. In contrast, in modern agricultural settings, we often see group selection. In such scenarios, selection focuses on the performance of groups of plants rather than individuals. And this distinction between individual and group selection is fundamental to understanding how evolutionary principles apply to agriculture and plant breeding. In contrast to the wild plants, such as the wild barley I just showed to you, modern crops such as cultivated barley are almost always cultivated in groups. You can see, this can be seen here in these examples of trial plots. For example, here is a plot of a trial. Here is another one. Each of these plots represents a group of genetically identical individuals. Keep in mind, they don't have to be genetically identical, but in this case they are. So for breeders and farmers, the focus here is not on the fate on the individual plants, but on the total yield of the plot, because they want to harvest. They are harvesting the whole plot or the whole field and then measure the yield. Of course, each plant contributes to the total yield, but the selection process considers the group as a whole, giving room for group effects, possibly by cultivating, for example, similar genotypes together. The total yield may be higher than cultivating the same number of plants separately from each other and then harvesting the seeds. And this is the definition essentially of, or one definition of the group selection, that there is a certain phenotype that applies only to the group as a whole. Here we could define this phenotype just as a yield per hectare. And this leads us to an essential concept in Darwinian agriculture: the distinction between individual selection and group selection. There may be traits that are only relevant at the group level, which cannot be selected for on an individual basis by selecting individual plants. For example, plant-plant interactions, so interactions among plants in a group, often play a significant role in group selection. Understanding and considering these interactions and their impact on group dynamics is crucial. We will explore this later in the lecture. Another important concept in this context is trade-offs. Trade-offs describe the relationships between traits where the improvement on one trait may lead to a deterioration in another trait. So to some degree traits are correlated and somehow interact with each other. For example, consider the trade-off between grain quality and biomass or yield. These two traits are not necessarily antagonistic, but they are related in a trade-off relationship. If you want to improve grain quality by selection, for example, you likely will reduce biomass or yield because improving grain quality requires resources that are then not available for the other traits such as biomass and yield. And in this figure, you see many other trade-offs that are interesting and relevant. I don't want to go into detail in the differentiation between the two different types of crops, lean and mean versus robust and competitive. For the moment, it's sufficient just to understand the basic principle of trade-offs. So if we aim to increase grain quality, it is often observed that the biomass and the overall yield of the plant may be reduced. And this has been documented in many different contexts. So essentially, the more resources a plant invests in improving grain yield quality, the less it can allocate to biomass and yield. As a result, the plant may produce fewer grains, but those grains will be of higher quality. And as the figure shows, in plant breeding, numerous trade-offs must be considered. This figure illustrates the difference between types of trade-offs, plastic traits and stable traits. Plastic traits are those that are more influenced by the environment, such as the availability of resources or other environmental factors. Stable traits, on the other hand, are more strongly influenced by genetic factors and tend to be less variable in response to environmental changes. For example, in many species, the allocation of resources to male versus female fertility is often genetically determined to a large degree. Some plants might consistently invest more in male fertility, while others might prioritize female fertility regardless of environmental conditions. Understanding these trade-offs is crucial for plant breeders, as it helps them to make informed decisions about which traits to prioritize based on the desired outcomes and environmental conditions. This balance between different traits is a key aspect of Darwinian agriculture and evolutionary biology applied to plant breeding because it defines the so-called ideotype, the type of plant that we are selecting for, that describes the optimal combination of phenotypic traits in a plant variety. A very important trade-off in plant biology is the growth defense trade-off. Investing in growth and yield requires substantial resources, and if a plant allocates a lot of its resources to growth, it cannot invest as much in defense pathogens because resources are limited. Pathogen defense always requires a significant amount of resources, and this is a key decision that the plant must make, whether to invest in growth and yield, thereby producing a large number of seeds and enhancing its fitness, or from a breeder's perspective, increase the yield that can be harvested, or to invest in defense mechanisms which are crucial for the protection against constant attacks by pathogens and herbivores. This figure illustrates various factors that play a role in trade-offs, particularly in the growth defense trade-off. First, we have environmental factors that provide resources for the plants, such as light, carbon dioxide, temperature, and humidity. These factors are essential for producing assimilates by photosynthesis, for example. Then we have the circadian clock, which determines the response to different day lengths and temperature and so on, and influences, for example, the length of the vegetation period and so on. And additional, we have the plant microbiome, which interacts with the plants and requires and also resources such as water and soil nutrients that also influence plant growth. The plants must now decide how to utilize these resources and how to integrate the external environment into its growth strategy. Should it produce a larger number of seeds to maximize offspring, or should it invest more in the internal defense? This decision is evaluated through various components of the plant and is mediated, for example, through plant hormones such as auxin or gibberellic acid, or proteins that measure environmental conditions and pathogen presence. If many pathogens are detected, signaling mechanisms may indicate that it's better to invest more in defense rather than growth, and the balance is switching into another direction. Managing these trade-offs and recognizing their implications is crucial for plant breeders and farmers, as I already mentioned. And the goal is to achieve an optimal balance, ensuring that both high growth rates and robust internal defense mechanisms are combined in a single variety. And this delicate balance is essential for maximizing the overall plant fitness and yield. Now, to achieve this goal, as breeders, we have a multitude of tools available for this purpose. I don't want to delve into the specific details, but I will cover a few key concepts. First, we have natural variation. This refers to the inherent genetic diversity found within populations of crop plants, breeding material, for example, and we can leverage this natural diversity to achieve a few goals, such as altering gene expression, change the protein activity, and so on. And by doing this, we can hope that we can improve the phenotypes that we want to improve. The main approach for utilizing natural variation at the moment is traditional crossbreeding, where we cross plants to combine desirable traits. Another strategy is rational design, where we systematically combine multiple disease resistance alleles into a single plant. This is called pyramiding, or another term is stacking, by crossing. And by doing so, we can significantly enhance the plant's defensive capabilities. So this is often used for resistance genes. But of course, we have to check that the yield is not compromised. Additionally, we can utilize crop wild relatives. These are more distant relatives of croplands, wild plants, because they often have high levels of disease resistance or genetic variants that promote high growth rates or seed yields, or provide resistance against drought. Conditioned expression involves controlling gene transcription and translation, which can be achieved by genetic engineering or other means. And the most recent advancement is the genome editing toolkit, which is a method, but it can be used, for example, to condition expression or to generate new genetic variation that can be used for breeding. And this technology allows us to precisely modify specific genes, and we can possibly fine-tune the investment of plants in growth versus defense, for example. So by employing these different methods, we have a fairly good toolbox available. We can effectively manage trade-offs and optimize plant performance for both yield and resilience. Plants live in highly variable environment. And this is a problem because plants cannot run away. So plants need a strategy to minimize the risk in a variable environment. For this reason, another important concept is what is called bet hedging. Bet hedging is just another word for risk minimization, meaning that one needs to hedge one's bets. So the risk is distributed between different entities, factors, strategies, resources, whatever, in order to minimize the risk of failure. This can be best explained by a key bet hedging strategy in plants, or target of bet hedging, which is seed germination. In wild plants, usually only a subset of seeds will germinate in the next year, and the seeds that don't germinate stay in the soil and form what is called a seed bank. They may germinate in the year after or 2 years after, when the weather conditions are potentially more favorable. Because it's often quite risky. Let's say it rains in the spring, the seeds germinate, and then a long period of drought ensues, and then all the seedlings are dying. And then for this year, there will be no offspring for a particular plant genotype. So this staggered germination is crucial for survival, let's say, of a plant lineage that contains certain genetic variants, because it's a good strategy to manage a risk of drought or other adverse conditions that could wipe out seedlings in a particular year. And by this, the long-term fitness of a genotype, of a certain genotype, evolutionary lineage, is maximized because the risk of germination is spread over multiple years. However, such a strategy is not practical for crop plants. Farmers buy seeds. Seeds are expensive, so they need all seeds to germinate in the year in which they are sown or in the next spring to achieve the highest possible yield. So they aim for 100% germination to maximize their harvest and ensure that all sown seeds develop into a crop plant that can be harvested. So crop plants are bred to germinate uniformly. Genes that control germination are selected, and the presence of seeds that might germinate in later years is minimized, because that's highly undesirable for farming practices. Farmers prefer consistent, predictable yields rather than dealing with plants that germinate years later and disrupt crop rotation schedules. But then, of course, in crop plants, the bet hedging strategy for seed germination is essentially knocked out, turned down, turned off. It's not existing anymore. So having this in mind, we can generalize the concept of bet hedging and say that bet hedging is essentially an allocation of limited resources in a way that minimizes the risk. Bet hedging impacts multiple traits within a plant. It can affect different tissues, stages of development, and interactions with other organisms. For example, a plant must decide how much to invest in defense against herbivores and pathogens versus how much to invest in yields or number of seeds or in fitness. So in highly variable and unpredictable environments, and bet hedging is a favorable strategy. and can be for this reason observed in many plants living in extreme conditions. In agriculture and for farmers, bed hedging is a highly valuable strategy. As demonstrated in this picture here, it is well known that land races of crops, so these are genotypes that have not been extensively improved by breeding in recent years and maybe hundreds or thousands years old and were generated just by the selection of the farmers, typically have lower yields, shown here, as modern elite varieties, especially under optimal conditions. However, as shown in this study and in several other studies as well, typically land races usually exhibit more stable yields. So both under favorable and unfavorable conditions, shown here, they show about similar yield, shown here, whereas the yield in the modern elite varieties is very different from each other, and under very extreme conditions, modern elite varieties may even fail. So it's a very good bed hedging strategy for a farmer to grow a land race that has a stable yield under variable environmental conditions, because then they can rely that in every year they have at least some harvest, some level of production, regardless of whether the year is optimal or marked by severe stress. This is particularly critical for farmers who are subsistence farmers, because they depend on a consistent harvest for their survival. So to summarize, bed hedging is a widely adopted practice in agriculture, and the challenge lies in how we can effectively apply this concept of risk minimization in breeding programs and other agricultural contexts. But by understanding and utilizing bed hedging strategies, we can develop crop varieties that are not only high yielding, but also resilient to environmental fluctuations, thereby contributing to food security and sustainability. And keep in mind, this strategy is essentially taken from a natural context. Plants do not live by themselves, so to speak, but they interact with other species and they live in a certain ecological niche. And this is addressed by the concept of the extended phenotype. An alternative term is also niche constructions. So plants can construct their own niche in which they are living in order to ensure that they can survive or that they have a higher fitness. Consider a maize plant, for example. Here we have a maize plant, and this maize plant secretes root exudates, which are chemicals that influence the organisms that live in the soil, such as soil microbes. Some of these exudates may be detrimental to certain bacteria, indicated in red, and beneficial to others, indicated in green. Beneficial means beneficial to the plant. These beneficial bacteria, for example, could enhance the maize plant's ability to absorb essential nutrients like iron, zinc, or phosphorus from the soil and thereby stimulate its growth. The intriguing aspect of this process and of the concept of the extended phenotype is that these root exudates can alter the soil microbiome. They can basically filter the soil microbiome by species, and this altered microbiome can persist in the soil, benefiting subsequent crops planted in the same soil. In other words, the maize plant modifies its environment in a way that creates a favorable niche in the future. So not only in a given year, but also in the next year, because the soil microbiome has been altered by the plants growing in the year before. And this is an extended phenotype. One of the interesting aspects of the extended phenotype is to think about which other phenotypes, interactions of the plants with the environment can be considered to be extended phenotypes. Quite importantly, the specific action of a plant on the environment, for example, which root exudates are being secreted, depends often on the plant genotype, because enzymes encode for this, and for this reason the extended phenotype is an evolvable and selectable trait that can be potentially exploited in plant breeding. Since plants live in a certain environment and interact with other species, they also need to evolve in response to their environment, because the environment is constantly changing. We can categorize the environment into two broad classes: the abiotic environment and the biotic environment. The abiotic environment includes climatic parameters which are currently changing rapidly. This category also encompasses new agricultural systems such as industrial agriculture, which relies heavily on high fertilizer input and extensive chemical plant protection, or organic agriculture, or pesticide-free agriculture, or precision agriculture, and so on. So the plants need to adapt to these changes. We need varieties that are essentially adapted to certain agricultural systems. And of course, this can be achieved by selecting superior genotypes or varieties that thrive under certain conditions. In contrast, biotic changes involve interactions with living components, living species. And these are primarily what we consider when we are discussing coevolution. For example, plants must evolve in response to pathogens that infect them and pests that consume them. So to survive, plants must co-evolve with these harmful organisms to avoid being completely devastated. However, the coevolution also includes beneficial interactions. Mutualists, symbionts are beneficial organisms. Because they live in partnership with plants and aid their growth. For example, certain soil microbes can help plants to mobilize iron and other nutrients from the soil. I already mentioned this before. Plant-plant interactions are another aspect of coevolution. Plants of the same crop species can interact in various ways. These interactions can be intraspecific, so occur between plants of the same species. Within this category, we can differentiate between interactions among related plants, for example, first cousins, or between unrelated plants. The nature of these interactions can vary depending on the relatedness of the plant involved. There are also interspecific plant interactions involving different plant species, and this has again many applications in agriculture. For example, there may be a competition between the crop and the weed, or we could have interactions in intercropping, where we, for example, cultivate a cereal and a legume together, and together they interact because the legume may provide nitrogen and the cereal may provide some other important function. And two varieties need to kind of co-evolve together, or two species need to co-evolve together in order to achieve the optimal yield. So understanding co-evolution is crucial for developing sustainable agricultural practices that enhance crop resilience and productivity in the face of changing environmental conditions. A very important concept introduced into evolutionary biology to explain the principle of coevolution is the Red Queen hypothesis. This concept is taken from Lewis Carroll's story Alice in Wonderland, where the Red Queen makes the statement, "Now here you see, it takes all the running you can do to keep in the same place." So we have to run all the time with a certain minimal speed just to stay in place. In the context of evolutionary biology, this means that hosts and pathogens, which are co-evolving, are engaged in a continuous evolutionary arms race. The host, in this case the plant, must constantly evolve and develop countermeasures to survive attacks from pathogens and herbivores. The Red Queen hypothesis highlights the perpetual nature of evolutionary change, where neither side gains a permanent advantage. Plants must continuously adapt to the evolving threats posed by their attackers to maintain their survival and reproductive success. And we can apply these concepts in agriculture and plant breeding. For example, understanding the Red Queen hypothesis helps us to appreciate the need for ongoing development of crop varieties that can withstand evolving pathogens and pests. And it underscores the importance of dynamic breeding programs that incorporate new genetic variation to improve plant resistance. Let me give you a few examples for this. I would say one of the most important aspects or relevance or examples for the relevance of the Red Queen concept in plant breeding is the breakdown of disease-resistant genes in crop varieties. For example, for barley mildew, which is caused by a fungal pathogen, is one of the most diseases affecting barley. Over the past decades, researchers have identified genes or genetic variants that provide a protection against mildew. Plants with certain genetic variants or alleles of these genes are then resistant to varying degrees to the mildew. And these genes have been introgressed or used in breeding of modern varieties. So when new barley varieties are being developed, they are shown here by the different names. They carry such alleles, and initially these varieties receive high scores for the resistance to barley mildew, indicating a strong protection. So these scores are usually given during plant variety testing and registration. However, over time the pathogens evolve and develop resistance to these resistance genes, so they can break the resistance, and then the resistance scores go down, and the plants that carry a certain allele are much more susceptible to the pathogen. And these varieties then are more difficult to cultivate and often are not sold anymore because the yield is strongly reduced. And this process, as shown here, occurs repeatedly. And because it occurs repeatedly, it also has been called boom and bust cycles. So the plant breeders really must run like the Red Queen in order to stay at the same place and constantly find new disease resistance genes, include them into new varieties such that barley cultivation in this particular case can be maintained. So we have to think about strategies that can mitigate these boom and bust cycles. One is to these strategies can be kind of summarized by slowing down the speed of this arms race, helping the Red Queen to run a little bit more slowly. And this can be achieved, for example, by deploying a diverse set of resistance genes within a single population or within a single variety. So pathogens have greater difficulty to overcome one resistance or several resistances, or to use pest management practices and so on. There are several strategies available. Another example is shown here with wheat varieties. It's very similar to what we have just seen with the barley. So we have different diseases of wheat, and they get initially good scores. These are the different varieties, but over time some varieties lose their resistance against certain pathogens because the disease genes have been overcome by the pathogens, the resistance has been broken. I mentioned before that crop plants are often cultivated in groups. So we could just say that plants are social organisms, or we force them to be social organisms because we plant them at higher density. So the question we now face is, which traits are required for plants that are cultivated at high densities, such as modern crop plants, compared to wild plants that may grow at lower densities or even alone without neighboring plants of the same or other species. And one can think about many traits that can be expected to be affected or change in this context. In addition to deciding, therefore, how much to invest in resistance versus growth, which strongly depends on plant density, other traits become also relevant, such as root growth. Typically, a wild plant, such as this one here, focuses or can focus or is selected by its own survival. And for this reason, traits that contribute to the individual plant's fitness are maximized. For example, this plant may grow its roots as widely as possible to cover a large soil area and extract the nutrients and water efficiently. Similarly, they may spread their leaves as much as possible to have an optimal or a high photosynthetic efficiency to produce as many assimilates from sunlight, water and carbon as possible. There's also clearly a strong selection pressure to maximize the number of propagules or offspring produced in plants. We could call it seed number, to maximize the seed number, and also to kind of bet one hedges to produce seeds at different panicles, for example. In contrast, modern crop plants grown at high density require different traits or modified traits, which is shown here. First of all, for these plants it's important to avoid competition with the neighbors. So just to stay within their own allocated space. So a breeder would select for genes that restrict root growth, such that the roots are not spreading too much. Then the growth of the leaves could be optimized by increasing the leaf angle, such that the leaves are not overlapping with the leaves of the neighbor and add some shadow to the neighbor, and thereby minimize photosynthetic productivity. And it is preferable for crop plants to invest in a single reproductive structure that is highly compact, highly optimized, that is can be more easily threshed. So modern crop plants have just a single large panicle, for example, or a single big maize cob that contains all the seeds, for example, and the yield that is of interest. Different crops have varying degrees of domestication and selection for social traits. And it's interesting to think about how much a given plant has been domesticated. And one can expect that plants that were bred for high density modern agricultural systems have a very efficient resource use and ease of harvesting. And for this reason, it's important also to understand these traits, how plants interact with each other, how they become more social, how they maybe sacrifice some of their individual benefit for the benefit of the whole group. So the total seed number of this individual plant may be lower than of this plant here. But as a group, there may be a larger number of seeds per area, for example. I just mentioned that plants have a different degree of domestication, and this can be very nicely shown here in this image from the descriptors of quinoa. Quinoa can be considered to be a semi-domesticated plant, and different genotypes of quinoa can be described by their growth habit. So here we have, let's say, a wild growth habit where the plant has multiple panicles, multiple branches, and requires a lot of space to grow. And in contrast, we have a domesticated form with a single shoot, a single panicle, and for sure this plant can be planted at much higher density than this plant to the right. And of course, it's very interesting to think about which genes control these traits. So the interesting question is now, beyond these obvious traits like the growth habit that a breeder can very easily select for, are there any latent or hidden social traits? And which traits fall into this category? We can consider, for example, exudates I mentioned before. How much exudate secretion is beneficial to the plant when it's grown in high density? This is usually not measured in breeding programs, so they are basically selected indirectly. But of course, if we can measure it somehow directly, then we can directly influence it, select for it in breeding. Yeah, so it may be advantageous to secrete certain exudates at high concentrations or others at low concentrations, and it's not really clear how one would select for that. Also, it's not clear how they are inherited and whether they are influenced by few genes or many genes. The key point is that there are likely a large number of so-called latent or hidden social traits that we don't know yet, that we are indirectly selecting by selecting just, let's say, for a high yield or for traits that are very easy to discover. But then we can ask, are they, how can we find those traits? Are they present in wild plants? Are they absent in domesticated plants? How can we introgress them? And so on. And but this is very interesting in the context of Darwinian agriculture, Darwinian plant breeding, because these are traits that usually are not on the agenda in conventional classical breeding programs. Another aspect is in this consideration is the role of competitive ability versus social traits. So by competitive ability, I would say we define the ability of plants to kind of maintain themselves and to be competitive against other plants. And this can be nicely demonstrated in this example of the tritrophic interaction in maize that has been described some time ago. So maize roots segregate exudates, shown here. These are compounds produced by the secondary metabolism of plants. These exudates attract root nematodes, and these nematodes in turn feed or invest insect herbivores that are in the soil in form of larvae stages, for example, larvae of the corn rootworm, which is a very important pest of maize. And by preying on these herbivores, their population size is reduced, and by doing this, the root nematodes prevent them from causing damage to the maize plants. And for this reason, the genes responsible for producing the exudates are selected by natural selection, and because this trait is beneficial to the plant. And this tritrophic interaction exemplifies how plants can influence the environment. So it's another example of an extended phenotype. And the interactions with other organisms to enhance their survival and productivity. Now we can ask whether there's a difference in this trait between wild plants and crop plants, or between populations of the crop plant. And in fact, such differences were observed. Here, this quote, it's a quote from a scientific paper, says that most North American maize varieties have lost the ability to emit E-beta-caryophyllene, which is an exudate, into the soil. And this exudate attracts the nematodes. So because North American maize varieties have been bred for the longest time and have been cultivated in a high density, there was obviously no need to produce this exudates and to attract nematodes. And by some mutations, genetic processes, this gene has been lost from the population. So the researchers thought that it would be interesting to find out whether by introducing this gene again, one can improve the attraction of nematodes per arm. And this is what's shown here. So the gene that produces this exudate was introduced using genetic transformation into maize varieties that did not have this gene. And then experiments were done to find out whether these genetically transformed plants attract nematodes. As you can see here, there's a clearly significant difference between the control and the transformed plant that can produce this caryophyllene. So here we have a very nice example that demonstrates first the potential of genetic engineering to reintroduce beneficial traits that have been lost in modern crops, and also showing how these concepts of Darwinian agriculture, using topics like extended phenotype, niche construction, can be applied in breeding in order to improve our varieties and improve protection against pathogens, especially since the maize corn borer, for example, and other insect pathogens become more and more important in maize cultivation because of global warming, because these pests have now more favorable conditions. So in order to understand why genes get lost in certain populations, we have to resort to population genetics. Specifically, just as one concept, the concept of the effective population size. This is a key parameter for understanding Darwinian agriculture and evolutionary process. And let me briefly explain why this is important. First, if a population has a small effective population size, there is a high loss of adaptive variation. In small populations, genetic variation tends to disappear, and much of this lost variation is adaptive. This occurs due to genetic drift and inbreeding, which are more pronounced in small populations. Second, we encounter the problem of maladaptation. While plants can adapt to certain environments in small populations, this adaptation often leads to the fixation of slightly deleterious variants. So these variants are not very strongly detrimental to the plant, but in some can lead to a reduction of the plant fitness or to the yield. And this process that fixes these deleterious variants is known as genetic hitchhiking, because a favorable mutation can bring along a harmful variation that is linked to the favorable mutation. Additionally, the efficiency of selection is reduced in small populations. For instance, the rate of co-evolution is slower in populations with a small effective population size. So they can more slowly adapt to the evolution of a pathogen or a pest. And this is a reduced efficiency, and this hampers the ability of plants to keep pace with evolving pathogens and environmental changes. The key mechanisms that operate at a small population size are genetic drift and inbreeding. Genetic drift means the loss of alleles by chance, and inbreeding essentially means that a large proportion of the variation becomes homozygous and therefore expresses recessive deleterious mutations. The theoretical background for this phenomena is provided by the nearly neutral theory of molecular evolution. Another important concept is selfish genes versus cooperation. It has been observed multiple times that certain genetic variants or alleles can maximize their own inheritance, possibly at the expense of the host organism. So these genes can be also called genetic parasites, for example, and they just use the host organism to improve their own propagation. In plant breeding, as an example, one important category of selfish genes are called segregation distortions. They exist also in other organismal groups, but for plant breeding particularly relevant are so-called mitochondrial male sterility genes. They are selfish because they cause male sterility. And these genes are encoded by the mitochondria, which are maternally inherited. So by making plants male sterile, they increase their relative chance of reproduction into the next generations, because they are transmitted by the female lines and all the resources, and basically increase the frequency of females in a population. And by doing this, these male sterility genes would enrich in a population. However, there are countermeasures to this phenomenon which reflect co-evolutionary dynamics or a red queen process. So-called restorer genes, which are encoded by the nuclear genome and are inherited from both the males and the female plants, can counteract the effects of segregation distortion and thereby keep them in check. So this is a classical example of a red queen process. Another example of selfish genes includes many transposable elements that maximize the inheritance by inserting copies of themselves throughout the genome. The concept of selfish genes was popularized by the British biologist Richard Dawkins, who wrote a famous book with the title The Selfish Gene. And he defined phenotypes or organisms as replication machines. So these are just vehicles for gene replication, of which genes tend to become selfish. This gene-centric view of life has been highly influential, but it also has faced significant criticism. The critical point is that selfish genes, due to their biased inheritance, may invade populations and therefore act against other genes promoting cooperation. And this could be potentially seen in traits such as restricted root growth or shade avoidance through higher leaf angles, which enhance plant interactions. It should be said, however, that the interaction between such selfish genes and cooperation social traits is an underexplored area of research, especially in plants. In microbes, such interactions are much better studied, also because it's easier there, but also in plants it represents a promising and wide open field for further investigation. Closely related is the concept of kin selection. Kin selection is a form of natural selection that is based on shared alleles. Essentially, it uses is used to explain the opposite of selfish genes, namely altruistic genes or altruistic behavior in populations. Kin selection stems from the observation that relatives share a higher frequency of common alleles due to having a common ancestor. They are related. And the British evolutionary biologist Bill Hamilton derived the concept of inclusive fitness from this observation. Inclusive fitness suggests that an altruistic individual, may sacrifice their own fitness or reproductive success to benefit a relative under certain conditions that are given in this equation here. Specifically, if the fitness cost to the altruist is less than the benefit to the recipient multiplied by the degree of relatedness, then kin selection can occur. So C would basically be the fitness of the altruist, or the cost to the fitness if the altruist becomes altruistic and is not selfish anymore. And if this is smaller than the product of the benefit to a recipient and the relatedness of the altruist to the recipient, then kin selection can occur. And this equation explains how altruistic behaviors can be favored by natural selection, and such genes conferring altruistic behaviors can be selected. In summary, kin selection helps to explain how traits that benefit others at cost to the individual can evolve, and emphasize the importance of genetic relatedness in the evolution of cooperative behaviors. Now you can ask the question, does it occur in plant? The answer is yes. There are several studies where this has been observed. For example, shown here, in rice plants and also other species secrete exudates from their roots that inhibit the growth of neighboring plants, a process known as allelopathy. This phenomenon is particularly important in contexts such as crop rotations and weed suppression without chemical plant protection, because this allelopathy can be used to suppress weeds. Interestingly, research has shown that plants that can recognize whether neighboring plants are relatives or non-relatives. And based on this recognition, plants can adjust the level of exudate secretion accordingly. And this differentiation indicates a form of kin selection because the production of exudates is costly. And if a plant detects a neighboring relative, both can collectively produce enough exudates to suppress weeds, for example, and thereby benefiting their whole shared genetic lineage. This topic is quite fascinating and highlights a sophisticated level of interaction among plants that is not yet fully understood. The ability of plants to distinguish kin from non-kin plants and adjust their behavior, such as the secretion of allantoin in this particular case, so of exudates, and to kind of have a negative effect on non-related plants, is a very interesting area of research and there's for sure much room to conduct interesting studies. Interactions between plants and other organisms can be beneficial, and there are many examples for this. Mutualisms are defined as beneficial interactions between species, and there are several examples. One classic example is the pollinator-plant interaction. A plant produces nectar, which the pollinator consumes, and in return the pollinator helps to pollinate the plant. Another example is the relationship between plants and mycorrhizae, because mycorrhizae assist plants in nutrient acquisitions, for example, iron mobilization from the soil and some other nutrients, whereas phosphorus, for example, and in return they receive carbon compounds from the plant that the plant has fixed by photosynthesis and then the mycorrhiza can use for growth. And we have the plant rhizobia interactions, where the rhizobial bacteria help the plant to fix atmospheric nitrogen and provide the plant with nitrogen and in effect also receive nutrients from the plant. The problem is that mutualisms are highly susceptible to cheating. For example, you can imagine an insect that consumes the nectar without pollinating the plant. So this mutualistic relationship is exploited. And there are species of bees, for example, that exactly show this behavior. And this raises questions, for example, about the stability of mutualistic strategies. How can plants manage or mitigate and in respect breeders who know about these relationships mitigate the impact of cheaters? Similar challenges can be observed in plant mycorrhizal interactions because some fungi or microbes might benefit from the plant without reciprocating with nutrient provision. Importantly, these interactions are strongly influenced by both genetic and environmental factors. So in agriculture, we can consider how to utilize them and enhance mutualistic relationships while minimizing the impact of cheaters. So if we want to utilize plant pollinator interactions, plant mycorrhiza interactions, plant rhizobia interactions, we always have to think about how cheaters can kind of disturb such a system and reduce the benefits from such interactions. But if we focus on these strategies, which is in the core of Darwinian agricultural thinking, we can improve crop performance and sustainability by enhanced cooperative interactions. There's a formal way to model and describe such interactions, and this is called game theory. Game theory is a theoretical concept that helps us to formalize these interactions and explore different strategies, for example, through simulations. Game theory is particularly interesting in frequency-dependent relationships or selection or evolution. For example, the strategy that an organism might adopt can depend on the proportion of cheaters in the population. So if a plant has an idea how many cheaters there are among pollinators, it can adapt a strategy. If the frequency of cheaters is low, one strategy may be effective, while a different strategy might be needed if the frequency of cheaters is high. So we can apply game theory in various contexts, such as describing host-pathogen interactions or the relationship between cooperators and selfish individuals. Another relevant context is the dynamics between honest signalers and cheaters. For example, in plants, a large and beautiful flower might signal to insects that there is also a good source of nectar which attracts pollinators. But it could also be a deceptive signal to lure insects that end up pollinating the plant but not receiving any nectar in return. So using game theory, we can describe and analyze these interactions, and it allows us to investigate the fitness of an individual and how this fitness depends on the strategies of other interacting individuals, whether they are of the same species or of a different species. So I have introduced quite a number of concepts. The question is now, what do these concepts mean for plant breeding and how can we actually utilize them? Classical breeding goals have successfully produced new varieties with a high yield quality and also disease resistance, because these traits are relatively easy to select for. Also, nutrient efficiency is becoming more and more important. It can also be identified in suitable field experiments. But our understanding of plant interactions with the environments and other species and microbes also has significantly advanced in recent years. So we can use Darwinian concepts, co-evolutionary concepts and so on in our breeding. In addition, we have now important political contexts that demand changes in agricultural practices, because the classical breeding schemes often depend on high input agriculture, fertilizers and plant protection, chemical plant protection. And this is becoming less and less easy and more expensive. And we need to reduce the input of pesticides and fertilizers. So we could think about how we can leverage species interactions, such as using microbes to help to overcome this dependency on the inputs. In addition, we are exploring the new uses of crops, such as biomass breeding for bioenergy production, biofarming, or producing specific chemical compounds. And also ecosystem services are becoming more and more important, such as buffering water to anticipate or to buffer drought phases, or providing pollen for pollinators to increase insect biodiversity. So we have a shift from a classical chemically driven, chemical input driven agriculture to a more resource efficient, sustainable agriculture that employs agroecological principles. And these principles often align with what we call Darwinian agriculture or evolutionary oriented agriculture. It's important to differentiate this from organic agriculture, because organic agriculture is possibly a more restricted set of agricultural methods that follow certain certification rules, and by some people it's often seen as only a marketing label that does not necessarily all the full breadth of agroecological principles. And agroecological agriculture is more broadly defined. This is at least one way to view it. Let's now discuss crop diversity and evolution as critical concepts in plant breeding. I previously mentioned population genetics and effective population size as important concepts. In order to fully understand the evolution of crops and their interactions with other species, we must always consider evolutionary principles. This includes genetics and genetic variation. It includes adaptation, co-evolutionary arms races, and the utilization of genetic variants by breeders. And these processes are influenced by different practices, processes that occur in nature and so on. And here are just a few of them listed. One is gene loss. I already mentioned gene loss before in the tritrophic interactions. Gene loss occurs very frequently in nature, and one question is whether we have a loss in phenotypic variation or a gain in phenotypic variation. There are examples for both. Then we can have, of course, new genes that are gained by new mutations. New mutations occur all the time. Again, these are very important processes. The question is the rate of new mutations limiting for a fast evolution or not? Then we have the the merging of individual plants by polyploidization, so new species, crop species, can arise rapidly that have a very high level of genetic variation. They may evolve fast. Breeders can select on them very efficiently and fast. And wheat is, of course, the most famous example for that. But new hybrids are created, new polyploid plants are created now all the time. And finally, gene flow is an important process, for example, between wild and crop species that can be done on purpose by introgression breeding. And this again allows us to capture useful genetic variation and traits caused by this variation that may be beneficial in sustainable Darwinian agriculture. In sum, we can possibly achieve our breeding goals much faster if we are well aware of these processes and use them for good purpose. After having introduced all of these concepts, we have now to think about the challenge for breeding. The challenge in breeding now lies in identifying which traits are relevant and determining how to efficiently identify and select for these traits. First, we need effective phenotyping methods for these traits. For example, I have introduced to you the example of root exudates. How can we efficiently measure the proportion or amount of exudates produced across a large number of genotypes? Furthermore, how can these traits be genetically mapped and what strategies for their utilization should we employ? If we can identify these social or latent traits, how can we select for them efficiently? Additionally, how can we develop low-cost methods to measure complex traits such as root size and root architecture that are also relevant in this context? Then, if we have many latent traits that were not discovered and were not being selected and may have even been lost in modern breeding material, how can we possibly rediscover them in genetic resources? These are really important questions. In the context of modern agriculture, which emphasizes reduced inputs, fertilizers, and chemical plant protection, we must consider the role of genetics in agroecology. Agroecology involves utilizing favorable interactions between species to achieve the goals that were previously accomplished through chemical inputs. So we have now to use ecological interactions and also genetics to achieve our goals, to be able to reduce our goals of sustainable agriculture. On a more practical level, which is relevant for our course, is the question which experimental designs can we use for complex interactions, such that we identify social traits or traits important for agroecology with a strong statistical power. This requires to some degree a more complex experimental design because we have to include multiple factors and multiple genotypes that are relevant. And for this reason, these are crucial aspects that require careful consideration. Closely related to this aspect is the question for the breeding methods. We have the classical methods such as population breeding, which is most likely one of the oldest breeding methods that exist. But we also have line breeding, hybrid breeding, and clone breeding as the most important methods in breeding programs that we have now. However, we can also explore Darwinian breeding methods that include co-evolutionary principles. These methods are designed to facilitate better adaptation of plant varieties to their pathogens and pests. And by maximizing genetic diversity in breeding populations, for example in evolutionary bulks, which consist of very wide crosses of genetically diverse material, and then let them co-adapt, evolve in their environment, we can accelerate evolution and adaptation processes. And there are some very interesting scientific studies that try to follow this paradigm. Land races and local varieties are particularly important for this purpose. And finally, also population varieties, where we again make crosses or just have populations where we select certain traits, focus then more on social traits, which can be highly diverse, where we use diversity effects within populations, because these populations are usually highly heterogeneous, in order to achieve our goals. Of course, we have then to think about which breeding method is most efficient in achieving our overall goals of a sustainable agriculture. The key point is now how to select for specific traits in breeding programs that align with Darwinian agricultural principles. We first need to consider the relationship between natural selection that occurs, let's say, in ancient materials, in land races, in wild species, and artificial selection by breeders, which is the predominant mode of selection in modern elite breeding. And in Darwinian breeding approaches, such as shown here, we often combine natural and artificial selection. So we give up a little bit of our selection to nature, to natural selection. We can exert a stronger selection pressure, also for different traits, but also we give up a little bit of control, because some of the traits that are selected under natural selection are actually deleterious for agriculture, such as a lower yield or higher plants that are more prone to lodging and so on. So we can integrate evolutionary principles into breeding, but we have to be always careful to plan ahead and think how we want to achieve this. Finally, I want to briefly talk about the further reading. Many of the ideas that I exposed to you are very well explained in the standard book by on Darwinian agriculture by R. Ford Dennison. It's a very good read. There are a couple of papers that have been published on the topic, actually quite a lot. What we will do is we will discuss this paper here, this concise introduction into the topic during this course. Thank you very much for listening.